Chytrid fungus is a type of fungus which can either survive by consuming rotting organic matter or by parasitizing a living host. Batrachochytrium dendrobatitis is a parasitic species of this chytrid fungus, which was discovered in 1999 and found to infect amphibians. BD was the first known chytrid species to parasitize vertebrates and has led to an amphibian biodiversity crisis due to its rapid spread around the globe, affecting a vast swath of species. The BD fungus can lead to a lethal disease called chytridiomycosis, resulting in massive population declines. The fungus spreads quickly, making it a huge problem for certain species, and in multiple cases has led to extinction of threatened species. The disease results in an abnormally thickened skin and disrupts nutrient and ion transport, which leads to cardiac failure and death. Some species, such as the American bullfrog, are immune to BD and therefore physically protected. However, they are still able to act as carriers of the disease and assist in the spread. Batrachochytrium salamandrivorans was first characterized by Martel et al. In 2013. Multiple amphibian extinctions have been linked to BD, but now this newly described fungus, termed B.sal, has been shown to be another causative agent of chytridiomycosis responsible for local extinctions of the fire salamander across Europe. Fungus found in lesions from dead salamanders sampled during declines were used to experimentally infect healthy salamanders who developed chytridiomycosis. BD was ruled out by PCR and qPCR. These species are closely taxonomically related, but B.sal has a lower thermal growth tolerance and dies at 25 degrees C, implying differential niche requirements. B.sal is suspected to be native to Asia and has in recent years spread to Europe largely as a result of the pet trade. Particularly high presence of the fungus has been detected in areas of Western Europe, where it has been detected in both wild and captive salamander populations. It's believed that the pathogen coexisted with Asian salamander hosts for millions of years, allowing these species to evolve tolerance, as demonstrated by the absence of any physical damage to individuals infected, such as skin lesions and mortality. While Asian Urodela populations have evolved resistance to the pathogen, European populations have experienced dramatic losses, particularly among fire salamanders, due to immunological naivety. After describing B.sal 5 years ago, Martel's group conducted an extensive phylogenetic analysis and determined that B.sal is a member of an until recently unknown lineage of chytrid fungus, which forms a clade with the BD species. From this analysis, the genetic distance between B.sal and BD was shown to be considerable, justifying the classification of B.sal as a unique species in its own right within the order Rhizophidiales in the phylum Chytridiomyceta. B.sal type chytridiomycosis is an aggressive degenerative skin disease causing rapid mortality generally within 30 days of infection. Salamander skin is a vital organ in maintaining homeostasis, the integument representing a critical route for water, oxygen, and ion exchange. Structural damage to the skin is therefore an acute physiological challenge. In infection, animals exhibit anorexia, ataxia, and apathy, and a rapid ulceration and erosion of the skin. Mechanistically, the virulence is a result of invasive parasitic growth of B.sal zoospores into keratinocytes of the skin. Each zoospore possesses a thallus, the fungal organ within which infected zoospores mature and develop, one thallus invading each keratinocyte, from which grow out processes called discharge tubes. These structures house cysts with maturing zoospores, and their growth and eruption causes necrosis of the keratinocytes, culminating in multifocal destructive lesioning and ulceration of the skin. This acute organ damage leads to a complete breakdown of normal ion transport, as well as presenting a substantial route of entry for other pathogenic microorganisms. This is in contrast to the pathology of BD-related chytridiomycosis, which typically causes epidermal hyperplasia and abnormal swelling due to overreplication of cells, and hyperkeratosis, a thickening of the cuticle, as opposed to erosive damage. Infection with either species, however, leads to a severe breakdown of transportation and regulatory processes with clear neurological and cardiac impacts seen in ataxia and cardiac arrest. BD produces a number of unique proteases which degrade elastin, an important structural component of skin, and given their close phylogenetic association, it's likely that B.sal uses similar enzymatic mechanics. It appears B.sal can also interfere with normal cellular replication and signaling processes, as Martel et al. Found unusual isolated islets of dividing keratinocytes clustered along the borders of lesions. Additionally, evidence has been found of bacterial colony growth on the thalli within cells of infected salamanders, which, depending on the species, can produce toxins or metabolites. B.sal zoospores have a relatively fast generation time of 5 days at 15 degrees C, contributing to the rapid onset of death as cells are invaded and ulceration worsens. B.sal is transmitted via direct contact between salamanders or by aquatic zoospores. Martel's experiment demonstrates transmission of B.sal across multiple Urodelin species as a result of direct contact, which provides potential for pathogen spillover. With regards to spores, B.sal makes a second type of spore which is hardier and rarely eaten by protozoa, and therefore can persist in water for months, making eradication very difficult. Spores can stick to the feet of birds, as seen in an experiment conducted on geese, allowing spores to be spread across continents. Additionally, the spores are also flagellated, which renders them motile and can infect soil up to 48 hours after an infected salamander has walked upon it. Infection can also be facilitated by organisms sharing the same habitat as salamanders, acting as carriers of B.sal. Midwife toads have been shown to be able to be infected and shed spores but remain asymptomatic, and high doses of the pathogen have been seen to kill alpine newts. However, low doses can render them infectious without death. B.sal will remain in reservoirs even after local populations of salamanders disappear, therefore any new arrivals will likely become infected from these carrier newts or toads. As with BD-related chytridiomycosis, the future predictions for B.sal are rather grim. B.sal is considered to have a very high invasive risk and has already been detected in populations in the UK and Germany. Conservation and science advisory groups in North America have urged for an immediate moratorium on the live salamander trade, given the high virulence, transmission potential, and mortality related to B.sal. Various surveillance systems and action groups exist for the early detection of BD and B.sal, which can expedite reporting and quarantine measures. But the fact that current-borne zoospores can be rapidly dispersed at considerable distance in waterways means even astute monitoring is unlikely to stop outbreaks downstream. For BD, various treatments which classify as applicable in the field, lab, or husbandry setup have been trialled, including UV exposure, dilute bleach solutions, Virkon solutions, and ammonium chloride, all of which were successful in killing zoospores. However, given the physiological role of amphibian skin, such harsh treatments may have other severe impacts on animal health. A more hopeful avenue might be the genetic route. Some frogs, such as Xenopus luivus, have been shown to possess immunity to BD infection, mediated by special peptides secreted by leukocytes. Genetic analysis, gene mapping, and candidate gene studies could then be used in an attempt to identify which genes might be responsible for conferring protection, and their protein products could be approached as either transgenic therapies or direct production of the protective proteins for application to infected animals.